I just did it myself. I didn't know how to give you permission.
Okay, fair enough. Okay,
And
Rachel.
then, all right, hold on. I'm going to add one thing here in the chat. This is my first time at doing a strategic roadmap presentation, but Anna has done this in the past and has found it helpful. So I've just added a link to RedCap, which is an anonymous survey she created. You all can fill it out while I'm talking or afterwards, and it's just on feedback on, you know, the particular parts of the presentation to make sure that we get better.
And Rachel, would you like people to hold questions or interrupt with questions, or how would you like to organize this?
I usually prefer to hold, but I can just because it's a little easier for me.
Yeah.
But
what if we pause and check on questions as we get through like chapters?
Yep, I'll do that through sections or Anna, if you can check the chat too in case anybody puts anything in there, that would be great.
Can you make me like co-host so when people join I can admit them and you can keep going?
I don't think I have I don't think I have an admission
okay.
Thing on there. I just let everybody show up. I don't care.
Okay, great.
Yeah. Okay. And everybody can see my slides?
Yep.
Yeah, will you make it? Yeah, there we go.
Yep.
Okay.
All right. Let me do some adjusting here. There we go. Okay. And for those of you who don't know me or I haven't had the pleasure of speaking with over the last several months, I'm Rachel Heilmann, along with Anna Falzer, and then Hilly's around here somewhere. She's our student who's made all these really pretty pictures in here. So she's our artist, and I'm one of CombinedBrain's postdoc fellows on staff here, and Anna is our chief scientific officer. So we've really enjoyed speaking with all of you over the last several months and appreciated how open everybody's been in conversations about your work and opportunities and the collaborative contributions that many of you have already invested in. And with your help, we've put together this overview of the research landscape to identify the most critical gaps to address and move the field of MED13L research closer to clinical trials and ultimately cures for these kiddos. The last 30 minutes of the presentation will be reserved for open discussion to get your feedback and recommendations and to address these gaps. For those of you not familiar with CombinedBrain, we are a nonprofit consortium led by over 60 patient advocacy foundations working with researchers, clinicians, and industry for treatment development. Our mission is really to expedite the development of clinical treatment options for kids with rare diseases by promoting translational and clinical research. Today, we'll be discussing the elements that you see here, with the end goal being that open discussion that I mentioned. Ideally, I'd like to discuss how we can come together as a community to further the treatment development agenda. Whoops, there we go. MED13L related intellectual disability, or MED13L RID, is caused by a variety of mutations, mostly large deletions, frame shifts, nonsense, and missense mutations on the 12th chromosome. The current prevalence is estimated to be somewhere between 2 to 6 in 100,000 births, with about 70 patients enrolled in the two registries and roughly just over 100 patients identified in literature. Like most rare diseases, MED13L RID is very heterogeneous in its presentation, with developmental delay and intellectual disability being hallmark symptoms that present in infancy. Some key physical features can include a broad, flat bridge nose and bulbous nasal tip, broad forehead, epicanthic folds, up-slanting palpebral fissures, an open mouth with a large tongue, and strabismus as present in this present ocular feature in this happy little baby right here. While first thought to be a disease characterized by congenital heart defects, the symptomatology, along with seizures, autism, and other behavioral manifestations, have only been present in about 20 to 30% of individuals, with seizures being a seemingly prevalent feature of a particular variant type, the missense mutation. Currently, there are no medical therapies to restore the cognitive or motor function. MED13L is a subunit of the dissociable cyclin-dependent kinase module comprised of cyclin C, CDK8, MED12L, and of course MED13L, and their interchangeable paralogs, MED12, MED13, and CDK19. MED13L expression is largely thought to be isolated in the nucleus in early embryonic and fetal development in the cerebellum and hippocampus, and is ubiquitously expressed throughout the peripheral tissues. The additional evidence shows that MED13L and likely MED13 are involved in regulating Wnt and hedgehog signaling, involved in neural crest differentiation, which is responsible for the development of the skeletal, cardiac, and facial structures. MED13L expression reemerges in early childhood and adolescence, and then appears again in adulthood and persists throughout life, although the contributions on this function with these spikes are not known. As I just mentioned, MED13L is largely thought to be located in the nucleus in early on development. However, in a recent publication from Drs. Hamada, Nagata, and colleagues, used a C-terminal-based antibody to show a change in nuclear isolation somewhere around E14 stage in mice, with persistent cytoplasm presence by P30 in differentiated neural crest cells. You can see here in this photo, starting on the left-hand side, marked by the DAPI staining, there's still nuclear presence of MED13L in these undifferentiated cells. They repeated this experiment in cortical neurons and showed an increased cytoplasmic accumulation as seen by the red stain. And finally, in hippocampal neurons, they noticed a co-localization, which is really hard to see on this photo, of MED13L with both presynaptic and postsynaptic markers. This seems to demonstrate something fairly unique to MED13L, which is its cellular localization changes with the developmental period, as well as its tissue and its cell type. The validation of this hypothesis may be key in unlocking additional areas for therapeutic targets. MED13L variants are largely thought to display a haploinsufficiency syndrome, as represented by this slide. The primary role of MED13L is a member of the CKM as a transcriptional activator and suppressor. If I can draw your attention here to the bottom left hand of this diagram, you can see that the mediator module shown here with a head, middle, and tail is connected to the kinase complex here in purple. If you look at the blown out picture of this, you can see the CKM or kinase complex is comprised of MED13 or 13L, MED12 or 12L, cyclin C, and CDK8. The dissociation of the kinase complex is what allows this mediator module to then bind to RNA polymerase II to initiate gene transcription. Additionally, there's evidence that MED13L is responsible for sequestering cyclin C into the nucleus to promote further transcription, whereas under conditions of stress, MED13L releases cyclin C into the cytoplasm. If we move over to the right-hand side of the diagram, in MED13L related intellectual disability and haploinsufficiency, this dissociation of the mediator complex is impaired. Additionally, excess cyclin C is then released into the cytoplasm and reduced transcription of genes necessary for mitochondrial function take place. Investigators here have shown that increased cytostolic accumulation of cyclin C results in increased oxidative stress, reduced mitochondrial DNA production, and increased mitochondrial fission. Oops. As mentioned in previous slides, the function and homologous nature between MED13 and 13L have been documented. This slide depicts the relative similar amino acid sequences between the two proteins. The reddish highlighted portions of MED13L sequence account for 52% positional similarity with MED13. Additionally, the domains of MED13L are thought to be similar, if not identical, to MED13. It's proposed that both MED13 and 13L have a defined N and C terminal with two LXXLL motifs, a leucine zipper, and a MED-PW region. The latter, which is predicted to trigger that conformational switch in the CKM that regulates the larger mediator complex activity I just described. The three Degron regions described here or shown here in green have not. Oh, is there a question? Nope. Okay. Have not been confirmed. However, given the pathogenic variants that have been validated for MED13 and 13L that have occurred around here, as well as the similar or the same amino acid sequence, this is a reasonable hypothesis. Structurally, as you can see here from a 3D model, there remains many unconfirmed and unknowns of the MED13L protein, as represented here by the amount of yellow, orange, and red highlighted areas, as well as the relative chaoticness around this well-folded center. Wrong way. There we go. Nope. While a large number of known phenotypical pathogenic variants are considered to generate the haploinsufficiency syndrome, there's a small percent of whole gene duplications that have demonstrated an overexpression. This is connected to a much milder phenotype characterized by more mild intellectual disability, motor, and speech impacts. Missense mutations account for somewhere between 20 and 25% of variants and have not been fully validated for cellular functional impact. However, in silico predictions from many of you here today have noted differences in a proposed dominant negative impact for a specific subset of mutations. Missense mutations in general are connected with a more severe phenotype with presence of seizures, more severe intellectual disability, and absent speech patterns present. The next two slides illustrate the relative unpredictability of pathogenic variants in MED13L RID. Here you can see the presence of unique point variants across the protein structure in a seemingly unpatterned fashion. The exception to this is this previously discussed missense mutations, which are clustered here around the Degron region in exons 15 through 17, which computational and predictive models have shown a dominant negative impact. Additionally, Dr. Hamada and Nagata just released a paper last week where they recently characterized a few C-terminal missense mutations with misfunctionality. They hypothesize that the mutations here in the serine dent region at the very end C terminal show mixed functionality, with one showing a primary dominant, with the two serine mutations showing a prominent dominant negative effect, and the threonine located right outside the serine rich region showing a reduced function, which more similarly aligns with the haploinsufficiency syndrome in both genotype and phenotype. There have also been some additional questions raised on if these point mutations in the C terminal presenting with a slightly less consistent phenotype could be connected to a different functional profile. An additional missense mutation worth bringing your attention to is the GLU332 GLY missense mutation located here right outside the C terminal, stationed near that first Degron. Again, this specific mutation may be worth characterizing for functional differences compared to the other point mutations present in this area. This is a pictorial representation of the large exome deletions and duplications that we've seen in MED13L RID. The deletions are expected to generate a classic haploinsufficiency syndrome, and duplications, as I've previously mentioned, are a relatively small percent of the overall documented population. This may be due to the much milder phenotype that presents, which has minimized an intense exploration of the genetic cause. If some of you are seeing yourself over here in an outdated headshot, it's totally true. You all are the amazing scientists and clinicians that the MED13L Foundation and the Syndrome Association in France have assembled to bring a wealth of knowledge, expertise, and awareness to this rare neurodevelopmental disease. Many of you have already established connections with each other, and the hope is today that new connections will develop as a result of sharing this strategic plan. This is a compilation of a draft conceptual model based on literature review, interviews with 2 caregivers, and discussions with key opinion leaders. The overall goal of a conceptual model is to describe symptoms of patients and impacts on patients and caregivers. Understanding the symptom presentation of the disease frames the collection of resources, the development, and potential future directions the foundations can take. The domains outlined here in bold display the documented symptoms present in the spectrum of MED13L individuals. This tries to account for virtually all symptoms reported in the literature, as well as any additional information that has been collected along the way. This radar plot was created to show a clearer picture of the hallmark features of MED13L RID. Both intellectual disability and speech deficits are the hallmark features of the disease, with moderate intellectual disability being demonstrated in approximately 80% of individuals. It should be further noted that just around 20% of patients are absent of speech, and they rely on gestures, noises, sign language, and augmentative devices when available to communicate. Other prevalent symptoms include motor delay marked by delayed milestones and specifically walking, hypotonia, and craniofacial abnormalities. The next several slides, I'll discuss existing drug development resource. Actually, I'm going to pause there. This is a good transition space if there's any questions. All right, I'll keep trudging along here. The next several slides, I'll discuss the existing drug development resources that the MED13L Foundation has collected. You will likely see some familiar references and names throughout this portion. Whoops. At the time of the presentation, there are 2 main sources for ongoing collection of patient-inputted information to establish a natural history study. Here in the US, the MED13L Foundation has engaged their base in providing 57 biospecimen samples and 32 active inpatient patients who are enrolled in Simon Searchlight's longitudinal surveys. In France, the MED13L Syndrome Association, along with Dr. Gamid, have established an active history collection for around 45 individuals. Both data collection platforms focus on autism as an underlying symptomatology and are accessible to approved researchers for in-depth data collection and analysis. In addition to the repositories mentioned, here, both Dr. Bain in the US and Dr. Gamid in France have participated in assessment of individual MED13L patients. Currently, frequency and type of data collection have varied between the 2 sites, but have included survey completion through Simons and Genida, physical assessments, EEG collections, and biospecimen collections when able. Ideally, alignment of these data elements across both sites could generate a solid cohort of MED13L afflicted individuals across the US and Europe. The next few slides will highlight the collation of available in vivo, in vitro, and other MED13L derived models available for ongoing research and support to expand the knowledge of functional, biological, and therapeutic impacts. current in vivo models are present in 4 facilities in various stages of iPSC development. Of note, fibroblasts already collected by Dr. Bain and Dr. Recupero have already been sent to Dr. Stritch at Rowan University for further validation of the mitochondrial mechanism of MED13L. Several animal models also exist for MED13L. Patient-derived mouse models are expected to be complete this summer at Jackson Labs, and ongoing efforts by Dr. Chad Gruder to generate tissue-specific flox models that mimic a variety of potential pathway impacts are underway, with some already existing evidence that show mice with heterozygous cardiac-specific phenotypes are both smaller and thinner than their counterparts. Additionally, zebrafish models with their comparable MED13L and 13 genes are being developed by Dr. Wanada's lab to validate previous insight from Dr. Utami that zebrafish demonstrate improper development of gill and facial features with knockdown of MED13B. A large deletion from exons will be used as the genetic model for simulating variants reported in the literature in both of these models. Additional resources include Dr. Strick's lab leveraging yeast models to explore basic functions related to the CKM, while Dr. Gamid has used CRISPR-Cas9 technology to create a knockout organoid model with ongoing characterization and strategies for further experimentation on rescue underway. The remainder of this resource section is dedicated to the types of treatment strategies for MED13L RID. The focus will be on repurposed compounds, as well as potential gene-based strategies with considerations for proposed biological mechanisms. The MED13L Foundation has heavily invested in identifying FDA-approved compounds that Nick already alluded to that can upregulate MED13L or reverse its pathological phenotypes through a variety of mechanisms. Seen here, they've partnered with Rarebase, who's used their functional platform and a library of approximately 4,000 compounds, including mostly FDA-approved drugs, and have identified up to 15 potential drug candidates that will warrant further testing. These compounds, a few of these compounds are shown here in green. The FDA-approved compounds are now being validated in patient-derived models with the next phase that including differentiating of those patient-derived iPSCs into neurons to test them. A more targeted hypothesis-driven approach to drug repurposing would be to investigate select compounds that is shown to be efficacious in other neurodevelopmental haploinsufficiency disorders. One compound, which is also FDA approved in pediatric population, is 4-phenylbutyrate. Here in the schematic from Dr. Kang's lab in recent paper, they've used 4-phenylbutyrate on patient-derived astrocytes with GAT1 variants. They demonstrated that GAT1 variants impair localization in the cell membrane, but after administration with 4-phenylbutyrate, there's increased GAT1 expression and functional activity at the cell membrane. These findings have provided a scientific rationale for several clinical pilot trials in neurodevelopmental diseases with presumed haploinsufficiency, as well as some suggested dominant negative effects. Its mechanism is not clearly understood, but reducing ER stress and histone deacetylation inhibition for improved transcription is thought to be one of many pathways. The options here on this slide have been compiled from the UCSC Genome Browser, the Comparative Toxogenomic Database, and my literature review for different options based on proposed mechanisms impact from MED13L provided by many of you in our conversations. This is a summary of mostly FDA-approved compounds that have shown an interaction with MED13L through a variety of different mechanisms. Further testing and evaluation in vivo and in vitro models should be undertaken to prove efficacy. The starred compounds that you see here are drugs that are no longer available in the US, but where similar safe FDA-approved compounds exist and could be tested in lieu of these. Also of note, I included MDMA or ecstasy, which has a well-known neurological impact. The rationale for including this is that other illicit substances have been FDA approved for clinical use in very specific situations. This is an additional summary of approved chemical compounds where the MED13L Foundation has partnered with University of Alabama, Birmingham, which used its unique AI computational published literature search. As you can see, there are some crossover with the previous slide, as well as unique contributions that could be further explored with validation. Again, the stars mean that these are no longer available in the US, but similar safe FDA-approved compounds are. The final types of treatment strategies are gene-based. Unfortunately, MED13L is a very large gene at almost 10,000 coding bases, making it very unlikely as a candidate for whole gene replacement. Additionally, there are unknown functional characteristics of the variant that can make gene-based strategies difficult. However, for those that are known haploinsufficiency, theoretical options do exist with the aim to prevent, rescue, or arrest MED13L related intellectual disability. Validation of these functional attributes of variants, as well as investigating the potential differences in the aspects of the 7 protein transcripts, may further open opportunities for gene-based therapeutics. One opportunity to continue to explore is the use of CRISPR rescue. And Dr. Strick's lab applied a new technology of CRISPR-Cas12a ICAP editing to mutant MED13L. To orient you here, row 1 is mutant MED13L, row 2 is the Cas12a rescued MED13L. Here in the first column in green, you can see a clear difference in the aberrant cyclin C in the mutant MED13L, as noted in the cytoplasm, with a relative more contained nuclear encapsulation of cyclin C in the rescued version. This is further transitions to the mitochondrial, where you see the red dots in a much more unpatterned, unpreserved nature here in the cytoplasm, again, in a more conserved nature in the rescued version. And finally, when you zoom in, you can see the dispersion of cyclin C in the green, interdispersed amongst the mitochondria, whereas in the rescued version of MED13L, you can see cyclin C resequestered here into the nucleus. Now that I've reviewed, I'll pause here again, sorry. Okay, I'm gonna keep on trudging.
Yeah, I have a question. Hi, everyone. Randy, maybe you'd be the best to address this. I'm just curious, that was very striking results that were just shown. What was the nature of the rescue here?
I thought I saw Randy hop on.
Yeah,
Oh, yeah,
he
he's
was.
here.
Is he on mute? Randy?
He's on mute. Yeah,
There I am.
there we
Okay.
go.
Can you hear me?
Yeah.
Mhm.
Yeah.
Yeah.
So the nature of the mutation or the rescue. What was the question now, Dylan?
Yeah, you can start with the mutation, but then yeah, how did you
Yeah.
rescue?
Yeah, it was a point mutation causing is a single nucleotide insertion caused a frame shift, exon 20. And so we just went in and corrected that exon. So we removed that extra nucleotide, and that gave us a normal protein. And so the phenotype we're looking at with this release of cyclin C, we wanted to make sure that it wasn't a secondary effect of the mutation, because sometimes you'll get a mutation and then the cell responds to that by doing something different. And by correcting the mutation and reversing the phenotype, it looked like the mutation was directly responsible for that phenotype. And so we just used the CRISPR protocol to make the correction. And it happened, the loss of aberrant cyclin C localization happened right away. So it was pretty quick.
Yeah, it's really striking. Thanks.
Mhm.
Thank you, guys. Any other questions? All right. Now that I've reviewed the background and summarized all the available resources, I will spend the remainder of this discussion highlighting the recommendations. This particular slide maps the process towards therapeutic development and serves as an index for the remainder of the presentation. The items you see highlighted in green have already been initiated. The text in yellow are items that are in process, and the red opportunities present areas for development. The blue text are some future development recommendations that may be undertaken to further the agenda. The recommendations I will be discussing are broken into 4 categories. Patient engagement projects are going to focus on increasing and capturing increased diagnosis rates, as well as empowering the community. Collaborative recommendations are to focus the attention on opportunities for the foundation to collaborate with others and clinicians and researchers to come together for a more streamlined approach. Either of these recommendations may or may not require funding, and recommendations requiring funding will be specifically called out. And finally, strategic decisions will outline some guidelines to continue to balance and build capacity. These recommendations for patient engagement to improve the time to diagnosis require no funding. I've taken the liberty of looking for MED13L as a gene of interest in both Invitae and GeneDx panels, and then also included some potential opportunities to solicit panel additions, an attempt to align the presence from one organization to another where that misalignment exists. I already know that Vanessa has taken some time to socialize the use of face match. This could be a powerful resource to identify and improve diagnosis through facial recognition. As the tool becomes smarter, the more data it collects and could provide insights to key facial features that are specific to MED13L RID. And finally, Beginnings, Find Out, Guardian, Ambit Care, and Probably Genetic offer free access to whole exome or genome sequencing in a variety of situations and across a multitude of age groups. In general, both MED13L Foundation and the MED13L Syndrome Association have done a nice job of engaging the community to enroll and participate in Simon's and Genida. It is key that this continues to be a priority to capture the symptom presentation and establish the longevity that is needed to build natural history studies. This can be done through organized social media plans, leveraging additional resources like trend analytics, publishing as a foundation, and creating educational materials such as the one-pager for educators, providers, and other members of the MED13L community, so they can continue to spread awareness and provide insight to the care needed for this population. Additionally, the MED13L Foundation already promotes the use of the CRID, and this should be continued to done so that resources are collected and tied to an individual throughout their disease journey. The final recommendation for patient engagement opportunities is the development of biomarkers. Excuse me. This can be done through the establishment of a foundation-governed biorepository and prioritizing collection of missense and C-terminal variants if lacking. And finally, leveraging those family conferences to make use of captured audiences, not only to collect biospecimens, but also EEGs. All of these should be made available in a central repository, linking those biomarkers through the CRID. As I've described earlier, all mutated components of the CKM have a documented neurodevelopmental phenotype. Interestingly, both MED13 and 13L have presented with the presence of cardiac defects in addition to the other common symptoms that you see here in the table on the right. This crossover provides a unique opportunity for leaders of the MED13L foundations to coordinate and co-fund basic science experiments around the mechanistic similarities and uniquenesses, and also investigating in potential compensatory mechanisms across the CKM. Additionally, identifying similar biomarkers and further coordination of natural history studies should be evaluated. An additional area for collaboration is between the foundations, scientists, and clinicians. The MED13L Foundation has been working very hard to ensure that biospecimens are accessible to anyone who would like to use them. An area for improved engagement is to host quarterly research symposiums with key opinion leaders to provide updates, review the newest literature or any other creative ideas that might lend itself to accelerated outcomes. Additionally, building the resources that Siemens and Genida have already provided for patient-entered information to create clinical centers of excellence with transparent data sharing will be key for trial readiness. Some proposed sites could include the US here with Dr. Bain, in France with Dr. Gamid, and attempting to engage Japanese experts should be explored as given the published literature of the small cohort present there. As mentioned earlier, the size of MED13L precludes it from traditional gene replacement therapy. However, given the strong likelihood that Degron position mutations are dominant negative, with only a handful of patients identified in the world, an application to NLORM for individualized ASO therapy should be considered. For the larger population, engagement with industry that utilizes gene agnostic strategies with dose control platforms could have the potential to capture the majority of the MED13L population. I provided a list of a few kinds of those technologies here, which may lend themselves to that opportunity. And then additionally, pursuing gene-specific options such as CRISPR base editing may be a good choice for those with very unique situations. The next handful of slides will discuss potential funding opportunities. One key area for clinical trial readiness is the ability to have outcome measures to assess. Funding the development of the final conceptual model to generate the unbiased profile of MED13L with caregivers, clinicians, and educators, and then to publish it as foundations will provide visibility and insight to the uniqueness of the disease. Additionally, validating the ORCA and ORTAS surveys in this population to assess both communication and toileting ability will be necessary given the impact that both these have across the population. And finally, leveraging these results to prioritize what matters to your community of families will be necessary to identify those common areas for targeted outcome development. Some additional areas for funding for outcome development would be to support the validation of an EEG as a biomarker, and by identifying a common pattern and using those family conferences as well as home collection opportunities to gather this data. Also, continue to advocate and provide incentives to prioritize the evaluation of the already existing Siemens and Genida data and publish those findings. When it comes to some basic science experiments, recommendations include developing an N-terminal based antibody that can differentiate between MED13L and MED13, as well as account for these early truncating variants. Continue to work to functionally characterize the MED13L patient-derived lines through quantifying protein expression. And finally, leveraging Adalorin as a read-through technology for nonsense mutations and expanding on CRISPR technology for rescuing phenotypes will continue to provide insight to some of those therapeutic interventions. The foundation has, in collaboration with many of you, built a large repository of animal models, patient-based, tissue-based, and conditional. There's an area of opportunity to test interventions for identifying a therapeutic window when an intervention should be given, and more importantly, where it shouldn't. Further exploration for a global conditional model should be considered depending on those outcomes. And as high throughput validation gets underway, it's reasonable to also investigate other compounds or combinations of compounds that focus on sequestering cyclin C to the nucleus and or target other known MED13L pathways such as Wnt. And finally, continuing to focus some resources on gain of function variants by exploring ASO technologies. And last but not least, the strategy for achieving these should be to prioritize and allocate funding throughout disease characterization, developing shareable research resources, and providing funding for both disease modifying and curative therapies, as well as clinical research. One way the foundation could do this is through a request for proposals to provide seed grants from the foundation to applicants focused on closing these gaps and collaborative efforts to meet those goals. Using an unbiased pre-review approach with anonymous scoring rubric, as well as hiring a part-time scientific officer, will be key to keep those grants fair and balanced. And with that, as Anna would like to say, this is my gang symbol for the MED13L community. Thank you again so much for allowing me into your lives and helping with this process. And we'll open the floor up to any questions. Anna, do you want to say anything or add anything before we do?
Yeah, I'll say, Rachel, that was really great. I think the way I would like to do this is really, since we have, you know, what I would consider the world's leading experts in MED13L right now on a Zoom call collectively, I'd really like to use this time to get feedback on, you know, the specific recommendations that we have found and sort of presented to you all. So we have discussion questions, but first it would be like, you know, depending on what is most appropriate for you, do you see, you know, in terms of the basic science projects or clinical research projects, anything right off the bat that you think should be added to our list of recommendations or excluded if they've already been done or characterized, et cetera. And we can go back to whatever slide since and we'll also give you guys all of these slides at the end too. So I think let's just open it up and see who has feedback and comments, and then we can sort of ask specific questions that we came up with. Okay.
Yeah, this is Randy Strick. I've got a question or a comment. So the compounds that have been identified by RareBase, they're going to be further validated. What exactly are the assays they're going to use to validate in the neurons being generated? What's the what are they going to be looking for as far as whether these drugs are potentially useful or not?
I think there's people from RareBase on the call, or I can answer, but I'll give
I thought there would be.
yeah.
Hey there, Randy. Nice to see you.
Yes.
It's Rudy here. Hopefully you all can hear me.
Yep.
So the primary endpoint of our experimental validation in patient-derived iPS neurons and controls is looking at the impact on the expression of the MED13L gene target. And we're also looking at a microscopy-based phenotype of the cells. So I'm not sure if what you've looked at might inform our, since you have a microscopy-based assay up and running, could be something that we could think about sharing some material with you to look and see if we can have the same effect on the mutant cells that you have in your lab.
Okay.
But our idea would be to really first primarily look at the effect on the MED13L gene target transcript. We also will be looking at additional production of MED13L protein. And then finally, we'll be looking for a microscopy-based phenotype as well.
Oh, okay. So what we look at two different things. One is the mitochondrial phenotype that was showed earlier. And the other thing is target genes for MED13L, we see misexpressed in our MED13L cell lines, patient-derived cell lines. So we have those assays up and running. If you do, one of the possibilities is particular compounds you think are most interesting, we would be happy to take a look at them in our assays as well. We have fibroblasts, they're not neurons, but we could even, yes, we could look at that as well.
We would be happy to share material, transfer material for those in vitro assays, and look forward to collaborating on that.
All right, cool.
I have hi, this is Kathy. Yeah. So I have two questions for Rachel. Now I have some comment. So first, you mentioned that the kids have intermittent spikes in expression. Could you clarify that? Yeah, at the very beginning.
Yep, sorry, I was trying to get off mute there. Yeah, so there seems to be just some intermittent spikes in MED13L expression, especially in the cerebellum and the hippocampus, right? So when you kind of look at the protein mapping, it seems to be upregulated somewhere around between up till 3 years of age, and then it kind of tapers off and then increases again around early adolescence. So it happens around like 10 and then tapers off again around, age 13 or 14, and then doesn't present again until adulthood. And so, or it's present, it's just not in as in a high of a presence.
So you're saying it's a protein upregulation in certain brain regions.
Yep.
Yeah, second question is related. Talk about the gain function is very clear, or is it just a speculation?
Say that one more time.
Or gain of function. So right now we'll talk about
the gain of function. Yeah. So so most of the work that's been done with the gain of functions has been computational analysis.
I see.
And then last week, so like hot off the presses, the Drs. Hamada and Nagata published cell-based characterization with one of the Degron mutations and then 2 serine mutations, missense mutations. So the Degron mutation looks to be a clear gain of function, and the serine mutations, one is looks to be a gain, and the other is mixed depending on the tissue or the cell line they looked at. So and I believe Dr. Gomid is on here. They're also looking at that and characterizing those specific a handful of specific missense mutations. So I think some of that work is underway there. There are a couple others that maybe are like the early missense mutation that might be interesting to characterize as well. So that just came out last week, where a few of those have been validated or characterized.
Yeah, so right now, is any patient iPSC cells available in your cell bank?
So yeah, so they're all right now. So iPSCs, no, they're all in the queue at Simon's for programming. And then there's, we have there's a dozen fibroblast samples over at Columbia that they're genotyping, and then I think we'll be sending to the MED13L repository here with CombinedBrain. So they're all in process. And then I think Dr. Gomid, which he can comment on, is working on. I think he has some iPSC derived models in France.
Yes.
Fibroblast.
Yes.
And there are 2 iPSC lines at Rare Base.
Rare Base, that's right. Yep.
Hello, hi, this is Chris Recupero. Just a quick question. Are there any other plans to reprogram iPSCs in addition to Simon's Big Q?
So we have talked about reprogramming some through CombinedBrain through Van Andel. Yep. A handful of other ones where we might be missing some samples, some specific variants. Yeah.
Dr. Gumid, I think you've been trying to talk. Go ahead.
It was just a comment. Yes, we are working on it. It's not so easy to identify gain of function because as you know, the function of MED13L is somewhat mysterious. So it's not easy to identify gain of function, actually. So we are still working on it.
Yeah. One thing that Rachel mentioned towards the end as part of the recommendations is to develop a more optimized MED13L antibody, which we've discussed with, I think, most of you on the call, and tried to get feedback and make sure we're designing it with a couple things in mind so that it can be used by the greatest number of scientists for MED13L, which is designing it so that it will differentiate between MED13L and then its homologous counterpart MED13, which, as Rachel showed on one of the sort of intermediate slides, there's a good deal of sequence alignment between MED13L and MED13. And then the other thing is that we're designing it so that hopefully it can detect human and mouse versions of MED13L. And then the last part is, which is probably most important for the foundation and scientists doing basic research experiments on it, is that it will bind. It's really focused towards the N-terminal region of MED13L so that it can start giving you an idea of what the impact, the translational impact is on these MED13L gene variants. So that is something that we have in process. We've gotten feedback from many of you on, whether we're doing that and selecting the most optimal antigen for that. And then that is something that will be available to anybody who, either contacts Rachel or anybody at the foundation, so that you can start measuring MED13L protein in your assays. Okay. Other questions about any of the basic research experiments that we have proposed? Rachel, would you mind going to sort of the in vitro side or the in vivo side? Doesn't matter. Thank you. Okay.
Can I just ask if there is something that you're seeing that's in the queue for Simon's that is not in line with what you're looking for your research, and it's a different variant that we have within our patient population that you're curious about, or you're looking for an iPSC for, please contact Rachel or Nick or myself, and we can, see if we can, fund that development. So just because it's not in the Simon's queue does not mean that we're not, going to push forward for it. If we have someone who's interested where it can directly impact one of our community members, we are, driven to help that process. So please, don't be shy about it. If you see something within, the whole roster of variants that we have, please reach out and let us know. We're just motivated to help everybody in our community. So thank you.
Well said, and we'd apply that really to all the tools. We really want to create a toolkit for all of you that's readily available.
Right, and Rare Base does have that one nonsense variant and one deletion variant in iPSC form right now that they are ready to share. If you're interested, we can get those over to you fairly quickly. It's a just quick coordination thing, and the foundation will support the fees to transfer them over to you. But again, if you have any other variants that you're interested in, Rachel, if you can, whenever you get a chance, pull up kind of the list, please reach out to Rachel, Nick, or myself, and we'll get that going for you.
These are the ones sort of in flight in terms of the queue, other than the ones that Vanessa just mentioned here at Rare Base. And then when I did my little lollipop diagram, I actually put in all of the samples that have already been collected by Simons. So these would be ones that, you know, that are just sitting there. And so, you know, we can ask for them if there's a specific mutation that, you know, any of you would like to look at, we can ask for them to add it to the queue. So they really just want some sort of researcher requesting it versus, you know, us just flooding the queue with cell lines. So, and I know I've exchanged some emails with Dr. Gamid on some, especially like this missense mutation here, on trying to get that to him so that they can, you know, further expand their experiments on that. But again, they're happy to do that with researchers.
Hey, Rachel, can we see if we can get some insight into maybe some more hypothesis-driven drug repurposing proposals, like specific compounds that we know that might be worthy of sort of pilot experiments, or any of those compounds that either, you know, the phenylbutyrate that we proposed or any of these that might be specifically prioritized over others because of their impact on, like, you know, the mitochondrial deficits that we know. Will you go back to your slide that has all of those?
Yep.
Yeah.
Yep.
Okay, so people who have worked, you know, Randy, I'm looking at you, Chad, like knowing the sort of cellular phenotype in MED13L, which of these, if any, do you think would be, you know, worthy of like maybe a very fundamental in vitro experiment in like stuff in patient lines?
Yes, I mean, needless to say, the foundation is eager to really test a number of these compounds, including many of these that are readily available and over the counter.
Hopefully your favorite is not ecstasy.
Apparently no comment from Nick on that.
So we're actually, this is Randy again. So we're looking at the antioxidant, and there's a couple other drugs that are involved in ramping up mitochondrial mass. Resveratrol is one of those, and even some of the other ones that are listed there as well. So we're going to use those in once we establish sort of a baseline of mitochondrial function for these cell lines we just got, the 12, we're going to use those drugs to see whether we can ramp up mitochondrial function by using these are already well-known drugs to do this. One of the things we did find when we first looked at the one cell line is that although mitochondrial DNA levels were low, as you indicated in your slide, they weren't mutated though. We didn't see any gross mutations in the mitochondrial DNA, which is encouraging because that may give us an avenue to expand the mitochondrial function within the cell simply by using one of these drugs. So we're cautiously optimistic that if we can establish a nice baseline, we should be able to, over a period of time, ramp up mitochondrial function. One of the things the iPS cells may give us, although it changes the metabolism of the cells a little bit versus what we're looking at, and especially like neurons, we don't know how quickly, like when we corrected the mutation, we don't know how quickly we could reestablish mitochondrial function. It may not, it's not going to be overnight, but we don't know how long it would take to do that. So that's one of the experiments we're very interested in doing, is if we can reverse the phenotype, how long does it take to get back up to wild type levels?
So you think, Randy, based upon the data that you guys have collected so far, presumably an increase in mitochondrial biogenesis will lead to increased function? Yeah,
so that's the hope we have. I mean, that's why when we looked at the mitochondrial DNA integrity, we were happy to find that it wasn't deleted or messed up, and at least in a general way. We didn't go into point mutations, but yeah, so that's exactly right. And
that was found after using sort of an antioxidant that could be theoretically readily available.
If there's
in a clinical setting.
Yeah, if you look in the literature for mitochondrial pathologies, which there's several known, there was a lot of work to try to basically do that, to plus to induce, there's the genetic, the transcription factors and the genetic switches that are involved in sort of monitoring cellular energy stasis and status and wanting to upregulate it if you needed to. We don't have to go into the details here, but yeah, so they established several drugs, although they weren't very effective because most of the pathologies had an issue with mitochondrial DNA, which you can't replace. So yeah, so there are several drugs out there. These, you've listed a couple that are well known to be things that stimulate mitochondrial function. The resveratrol is sort of in that.
Have you tried yet to measure changes in actual MED13L, either mRNA or protein expression? So you're rescuing this phenotype. Have you looked to see if resveratrol or whatever the antioxidant you're using actually directly impacts MED13L expression, or it's just sort of fixing downstream?
Yeah, no, we wouldn't anticipate that it would affect MED13L. I mean, we have no reason to think it would. Well, we would look, but in general, what we would look for would be, it's a downstream effect. So it's dealing with the symptom, not the cause. So yeah, but that would be something you could do, especially if we get really good antibodies.
Yeah, here's hoping.
Yeah.
Okay. Any other ones on this list, or maybe there's like some on these lists that overlap with the, the compounds, or this least class of compound that Rare Base has found too, would be ones that I would prioritize if they're showing up on multiple lists as hits. That would be one that we could fast track in like an academic setting to try that specific, those specific targets.
One thought I had, guys, and Randy, just in response to some of your comments, if you're really looking for something to juice mitochondria quite potently, I'm not sure if a peptide would be compatible with your assay, but if you haven't looked at elamipretide, it's not something we've tested either to look at its effect on gene expression, but it really, elamipretide, SS31 or Bendavia has a few names, and that one is clinical stage molecule from Stealth Biotherapeutics. It might be worth a look. I think that peptide is available from compound suppliers if you don't already have it.
Yeah, no, please send me the information on that, or names or something. That'd be great. Yeah, because we're looking for, there's several already established drugs out there, but there's something that's especially useful or effective, then yeah, we'd be more than happy to hear about it.
Omid, would you mind putting the names of those compounds you just said in the chat? I tried to spell them and I think I have failed. That'd be cool. Yeah, okay, great.
So I'll just point out, just to support Randy's on the in vitro stuff, in the cardiomyocytes, at least when we knock them out, it's a mitochondrial phenotype. The transcription levels, the mitochondria total goes down as well. That's cardiac specific, but it totally supports the idea that this is at least has an overall phenotype as well.
I would have a question. Yes, we are discussing about mitochondrial dysfunction, but the patient do not show features of mitochondrial dysfunction. How can we explain those things, that there is a mitochondrial dysfunction, probably at the basis of the disease, but why patients do not show features of, just like patient with POLG mutation, or mutation in causing mitochondrial dysfunction? I don't know if somebody has the answer of this question.
Yeah, I can speak on that a little bit. I mean, the POLG mutations, when you're talking about something as severe, you're almost completely eliminating mitochondrial function. This is something much more subtle. And what we're seeing is that at least in the one cell line we're looked at, and we have some evidence in our newer cell lines that we're seeing a similar phenotype, is that the fragmentation of the mitochondria is in itself does not inhibit mitochondrial function, but persistent fragmentation does cause over time a loss of mitochondrial DNA and some mitochondrial function at that point. So what we're looking at is sort of it's a mild mitochondrial defect, but the sensitivity of neurons and things like that to mitochondrial morphology, fission, fusion, and function is pretty well known. So neurons are particularly sensitive to changes in mitochondrial function. So we don't look at this as a major loss of mitochondrial function, but it's enough. And that's what we're sort of focusing on.
Okay, thanks. Another question, did you try just like coenzyme 10 or something to rescue the function of the cells?
I'm sorry, using what?
CoQ10. I don't know how to say in English, but it's the coenzyme.
Oh, CoQ10.
Yes, thank you.
Yes, right.
Sorry, coenzyme 10. Sorry.
Yeah, so no, we haven't gone into that in great detail because we wanted to validate that this was actually something that's going to be important first. So like I said, all of our data was done on one cell line, and so that's, even though the statistics are really strong when you got one and you get the phenotype. So we're waiting to see how this works out with these other cell lines that we have. And once, like I said, once we establish a baseline that we're comfortable and confident that we've got something to look at, then those types of drugs and like Omid just sent us, those types of things we'll be definitely looking at. We tried the correction when we corrected the mutation in the fibroblast cell line. Unfortunately, those are mortal cell lines, and we sort of ran out of they senesced on us. So we weren't able to follow the correction very long before they quit growing per se. So we weren't able, with the genetics correcting it, seeing a reestablishment of mitochondrial function. So but we're hoping that now that we know what we're looking for, we'll be able to do a better job with that, and maybe the iPS cells that we've been talking about will also give us that experimental reagent that we need to address this question better.
Randy, do you have an estimate for when you may get readouts from your dozen cell lines? Or
well, we have some readouts, but we're waiting. The problem we ran into was a proper control, and so we didn't get. So we have lots of mutant cell lines, but we don't have a good control cell line that matches age and very well. So we finally got a hold of one, and it's coming now. So I'd like to compare the control first before I go into it, but we've looked at 3 or 4 cell lines so far.
Well, if the foundation can help you accelerate that, we'd be happy to.
Okay, yeah, so that, getting a normal cell line from a child that age is something that's, it's not something that's around. Very, very
You want like an unaffected cell line, or would you rather us think about making an isogenic line for one of the ones that you have?
Well, I mean, the isogenic would be fine. I mean, that would be ideal. But again, we're running into issues with, by the time you develop that, you've got a lot of generations. And so we're trying to stay away from immortalized cell lines right now.
Okay.
But so if we even had a young child's fibroblast cell line, male or female, would be helpful. We got a cell line from ATCC. It's on its way. It's as good as we could find. So.
Yeah.
Randy, we have a lot of motivated, siblings in our families too, that are more, well, the parents are more than happy to consent to them giving. Because, like myself, I have 2, like a 12-year-old and a 14-year-old. Katie has, 2 older boys. Lacey has, and then Ella has a twin sister. So, I mean, just ask. And these are the things that I'm hoping all of the scientists here are hearing from the foundation is just reach out, let us know what you need, what you want, your wish list, and we will do our best to make it happen. So yeah, so we can work on that, Randy. I'll email you separately. Maybe we can coordinate getting some fibroblasts of the siblings over to you.
Okay, so so let's do that. We'll see how the cell lines we have should be arriving anytime now. So
Okay.
I was also going to suggest that, Randy, when the next group of patients may come to see Jen at Columbia.
Mhm.
I don't know when that will be, but we could potentially coordinate that and
Yep.
send
Yeah,
some things for
I-
that, if that's possible.
yeah, I thought that we were sort of had that coordinated for the first batch, but apparently that didn't happen. So,
There's, I know, Nick and I are, you know, like 30, 45 minutes from Columbia. So we have 3 kids right there that we can come in anytime, Chris, to bring in these siblings, and then.
Yeah,
you know,
just
we can-
email me and Jen, and we can coordinate together. That'd be great.
Okay.
Thank you so much.
Sorry, Nick, I'm offering up Sophie.
Yeah.
Not asking.
I want to pivot just a little bit just before we run out of time, because I want to ask some questions about the in vivo work we proposed. So Rachel, can you switch the slide? So there is a robust, you know, Chad has developed several more than several mouse models right now. And I think one of the experiments that I want to propose that it should be, you know, a relatively high priority is that, you know, given that MED13L is expressed into adulthood, it's considerably different than the other combined brain disorders we have worked with, which is like it's almost exclusively expressed during development. So that to me means is at least a preliminary evidence that the therapeutic window for intervention for MED13L may be kind of large. Doesn't have to necessarily be corrected during development, given that it's expressed all into adulthood. Now, that's very hand wavy right now because no one has looked at that. But one of the things I think might be beneficial is to get a global conditional model, which I don't think exists, but Chad, I hope you say that I am wrong if I am. Since MED13L is ubiquitously expressed, to be able to do those therapeutic window experiments, you know, if you correct MED13L sort of in adulthood, can you fix, or what phenotypes can you fix in an adult rodent as an adolescent, etc. Chad, do you have any thoughts on that?
Yeah, so I'm at a swim meet, so there's a lot in the background. I apologize. But we do have the germline deletion, heterozygous germline deletion that we can do those studies. I've done the RNA-seq in hippocampal from 6-week-old mice, which are young adult. So that's for that. The as far as conditional deletion, we had the flox mice. It's just a matter of what tree line we want to use for that conditional deletion. It's inducible free.
But can it be globally expressed? It's like a global cree, or it would be knocked out in a specific tissue or a specific cell type. That's what I was thinking is like, in order to mimic the human disease, do you think there's any credence to doing something that would be a global conditional model? That's
global condition.
Do you know what I mean by that?
Yep.
Okay.
To see if it's developmental.
Yep.
Dependent or not.
Exactly.
I'd love to go the other way, to be honest with you, but
no. What I mean is like not to knock it in, but to fix it, to correct it.
Oh, correct it in adult. Oh, no, we don't have that. Sorry.
Yeah, that's what I meant.
That's
It's like
where David Liu or somebody would come in because base editing with the new mouse models that are being made.
Yeah.
He could do that time window, but no, as far as like what we have, you know, once you flox and delete, you can't add back with the tools we have right now.
Yeah.
But like I said, with the mice that are being generated at Jackson Lab, at least 2 of them, you know, that's the point of humanizing them to do the base editing and to correct those. And then apparently I don't know that system, but he could do the time course.
Yeah. Okay.
But I will, to the point, and we haven't done it in neurons, but we've looked at heart and liver. The expression, as Patty, I believe, was asking about, it does, or and Rachel pointed out, in mice versus mouse, the expression levels aren't even through development. We see, you know, high in embryonic development and then down and then back up. late in older mice. So it does vary. The expression levels, at least in the heart and liver, do vary over time.
Yeah.
I would be surprised if they don't in the brain as well. But that's something that is on our list to do a developmental time course.
Yeah.
That's a little snapshot, but
Awesome. No. Okay, cool. Yeah, that's helpful.
Caddy's asking, is the mutation knock-in mouse model available?
conditional knock-in, no, there's not.
Yeah. Okay. So I am going to pivot one more time because there's one more question I really wanted to ask before we ran out of time, which was a clinic, like maybe addressed to some of the clinicians on the call, which is what we're thinking about. In a couple months, we're going to maybe have a cohort of MED13L kids in one spot. A two-party question. One is, do you know, is there any preliminary data to suggest that EEG might be used as a biomarker for MED13L as we're sort of gearing up for the first round of pilot clinical trials? And two, what would be the feasibility of collecting EEGs at this upcoming family conference? I saw Reza on the call before.
Sorry, the question was we should collect the EEGs.
The question is, do you know, is there any reason to believe that an EEG could potentially be a biomarker for MED13L? Jen, I don't think Jen is here. So go ahead.
Sorry.
No.
I was just going to say, for context, we're gathering families April 30th, the Sunday before the Global Genes Conference in Philadelphia begins that Monday. And as we build up our critical mass, we'd be keen to engage with researchers if there's anything that you find valuable, whether it be testing or biospecimen collection or whatever. Thank you.
I do want to say, I do think that the families that are have raised their hands already to go, I think those are the same families that participated in Baltimore. So I think a lot of them had their EEGs done by Dr. Bain already. But if there are any new ones, maybe we can coordinate something.
Great. Yep. Come on.
I don't know. In France, all the patients do not have a systematic EEGs. Just if they show features of epilepsy. Not all the patients show epilepsy. Just about a third of patients, especially patient with missense mutation. But I don't know. Probably doing systematically EEGs will be good for the patient. I have no the answer on the question. But it's not something we do systematically here in our. There is no guidelines for doing this now. But I would be interested by the Dr. Bain answer on this question. What's her experience?
Yeah, I don't think.
I think we're waiting for the readouts for that, right, Anna? We're waiting for that. Yeah.
So we don't have to limit ourselves to EEGs. I think I would just take a minute to think of, based upon your own research, what would be helpful for us to collect.
Yes, to collect will be to do assessment of the the cognition from the beginning and through the. I don't know, through the growth of the patients, to do assessment of the cognitive capacities, to see what are the real difficulties, and it could be a readout if someday we have an essay to test, and to see the increase of the cognition capacities of the patient. That's something that could be really interesting.
Well, do you have a favorite assessment tool that you use, Jamal?
I have no favorite.
Yeah. Okay.
Ask Rosalind Combe could not be here today, and probably she could help us, but we do not have a specific assessment.
Yeah, Jamal. In the States, all of our children that have entered into school, they're school-age children, we all have Vineland tests that are done. And so I do know that that's something, like I've emailed mine to Reza before. I do believe that we could get a collection of people to, you know, black out and maybe just identify them with CRID and then have them, I don't know how we would like send it over to you, but we could probably figure out something. But the violins are very easy. They're standardized tests that, you know, the kids have to get periodically, and it's an excellent way to measure. And Simon's actually collects that for us too.
This collection of Vineland or other scale will be really, really helpful. Actually,
okay.
know exactly what are the precise difficulties of the children, the language, and I don't know, the visual, spatial ability also. That will be really interesting and helpful.
Yes.
Yeah.
Yeah. Hello, everybody. I also completely, I just wanted to agree with Jamal because the EEG, I don't also know what has been the hypothesis to really work on EEGs significantly, because among the patient total in different cohorts, less than 20% have seizures more than one time. It's really less than 20%, and type of seizures are very diverse, which somehow seems to be not something which is kind of systematically associated with the syndrome. That I also would rather go for clarifying the domains of the cognition by different tests like Wechsler or Venland and others. So I also think the emphasis on that is more than EEG, but neurologists must comment if really they can pick up something extra from the EEG than seizure activities.
One thing we don't know for sure, but we do have a sense that EEGs tend to be abnormal, even if they are subclinical in their presentation and there are not seizures present. And so, you know, as we think about clinical trials, cognition would be fantastic, but it takes so long and it's such a difficult measurement. If there were a way to correlate cognitive ability or other things with EEG performance in a way that we could get a more rapid biomarker, that would be
Yeah, that's true, but also, I mean, then EEG also needs to be, I mean, you cannot only rely on one-time measurement, right?
Yeah.
Probably it would be needed to be maybe longer monitoring or several times. So yeah, eventually could be effective, but I think if we have better cognitive assays, perhaps it's more useful also. I'm also in the direction that Jamal was mentioning.
I see Dr. Tranfaglia has a question from Fragile X Foundation.
Yeah, hi everybody. I'm the medical director of Fraxa Research Foundation, and we've been confronting all of these issues in Fragile X for more than 25 years now. So I just wanted to respond specifically to the issue about EEGs. Fragile X is similar in that we have about 20% of the males with Fragile X who have seizure disorders, but there is a distinct EEG signature in Fragile X that cuts across all the subjects with Fragile X, and it correlates pretty well with the severity of clinical involvement as well, just intellectual disability, behavioral impairments, and all the rest. So the rationale for doing EEG is that each disorder probably, I would say certainly, has a characteristic EEG signature. Also, sensory processing problems are a big issue in all autism spectrum disorders, and it appears to be one in yours. I'm not clinically familiar with MED13L, but just about everybody with any autism spectrum disorder has a problem with sensory processing. And so event-related potentials, like auditory evoked potentials, for example, can be a really useful instrument, and it is something that is druggable. It's drug responsive, and one of the questions people have when we talk about EEGs is, well, it's all well and good to develop this kind of signature, but is it actually going to respond in the course of a drug trial? And we already have evidence in Fragile X that indeed it does. So these are the things that you can investigate using human, let's use the broader term, human electrophysiology, not EEG. It's done via EEG. And in fact, when you put on that hairnet with the EEG electrodes, you can just have little headphones built into it so that you can do the auditory evoked potentials at the same time. And you can do repeated stimulus and look for the rate of habituation to repeated stimulus, which impairment and habituation is a characteristic of a lot of autism spectrum disorders, and it's pretty pronounced in Fragile X. So if you haven't looked already, that's all really important stuff to look for. And I would just add as one final point, it's really useful if you can have all of the data processed and analyzed in a single facility. So if you have somebody who is, you know, the EEG person for MED13L, that would be really helpful, because as we all know from, you know, working with scientists, everybody has a different way of doing things. Well, guess what? Electrophysiologists all have a different way of doing things too, and they have a different way of analyzing the data. And our folks actually analyze the most electrophysiology data using the exact same computer algorithms that they use to analyze the human data, so we can do a direct apples to apples comparison.
Yeah, that's a great point. I think we have a pretty sizable cohort of EEGs right now within one investigator, Jennifer Bain. And the goal might be to see if we can add to that at the meeting in April in Philly, maybe, and then start looking for, you're right, not necessarily detection of seizures in kids, but a distinct pattern that can be used as a biomarker of, like, you know, clinical trial efficacy. Any other last thoughts before you guys all get to go on about your day? No?
If no one is going to speak, I just want to make note that at this point, nobody knows if there's like a timeline for these kids, if there's like a shortening of their lifespan. I think that Nick and I have spoken to a few of you and told you that one of our oldest members passed last year. She's 35. And now one of our oldest members that we know of, she's 23. She's starting to have a lot of medical issues. They don't know what's going on. She's in Canada. They've done everything, no changes in MRI, no seizures, but she was super verbal, you know, was able to text with her telephone and, you know, all sorts of stuff. Very lovely lady, but now just out of kind of nowhere, she's become kind of non-responsive, and they don't know what's going on with her. So we don't know what MED13L has in store for our children, but we are just so grateful as a foundation and as parents for you guys, for your dedication, your inquisitiveness, your research, and your help for our family. So I just wanted to say thank you so much, because every time you guys think about MED13L and think about how you can help it or how you can learn more about it, you're helping us. So we appreciate it. So thank you.
Yeah, thank you guys, everybody, for your time and your feedback. If you do want to fill out the survey, the REDCap survey, and say things anonymously that you were too scared or didn't want to hurt our feelings to do it, please do so. We both have thick skin, and the whole purpose, like Vanessa just said, is to help these kids. So we will send out slides and the recording for this if you wanted to watch it again, maybe if you had trouble sleeping at night or something. But we will reach out to you probably individually to maybe follow up on some of the comments that you made, and we will be in touch very soon. So thank you all for your time. We know everybody's busy. Thank you guys.
Thank you so much, everybody.
Thank you so much, guys.
Special thank you to CombinedBrain and Rachel.
Bye. Thanks, guys.